Spirogyra is a green algae with more than 400 species, most of which are found in freshwater areas. Experiments have been conducted to test the effects of acid rain on algae by varying the pH. Algae is the basis of many aquatic food webs, and changing the pH can lead to its death. According to a study by Hoffman and Lusova, the diversity of algae increased and the densities remained around the same when a higher pH was compared to a lower one. The purpose of our project was to measure how algae would react to raising and lowering the pH. We hypothesized that adjusting the pH out of the natural range would kill the algae. Campbell gathered 10 test tubes and used a random number generator to label the tubes Raise pH 1, Raise pH 2, Control, Lower pH 1, and Lower pH 2, duplicating each test. Using a pipette, Jacob added 6 milliliters of the algae culture to the test tubes. Campbell created a soil-water mixture of Nature's Care organic garden soil by Miracle-Gro and added 3 milliliters to the algae. Using the pH meter, we recorded the pH of all algae samples. Using a micropipette, Katie added 0.5 microliters of 6 molar hydrochloric acid to lower the pH and 6 molars of sodium hydroxide to raise the pH. On Tuesdays and Thursdays, except November the 23rd, we checked the pH of algae to measure the changes over time. On the 17th, we measured the pH, added 3 milliliters of the soil solution to the tubes, and then readjusted the pH on the test tubes to match the pH before adding to the solution. Once we finished measuring pH for the experiment, we dried the algae to weigh the amount that had grown in each type of environment. The algae was placed into a dryer at 100 degrees Celsius for 8 hours. Once the time was over, we masked the algae contents of each test tube. In Figure 1, the graph shows the measurements of the algae samples raised by 1 pH. In Figure 2, the graph shows the measurements of the algae samples raised by 2 pH. In Figure 3, the graph shows the measurements of the algae samples lowered by 1 pH. In Figure 4, the graph shows the measurements of the algae samples lowered by 2 pH. Figure 5 displays the mass of the dried algae. Figure 6 displays the pH of each day measured.
Our data showed that the algal samples usually stabilize to around a pH of a high 7 to 8. Spirogyra are frequently used to clean up waters due to its ability to absorb ions. This method is being used to absorb the chromium ions produced from tanning industries, Chacha et al., and to clean waters of heavy metals such as copper-2 and lead-2, Chang and Lee. The optimal pH for the absorption to occur is 5. The pH cannot be too low because the hydrogen ions will compete for absorption, but it also cannot be higher because the alkalinity obscures the absorption of the ions. Our hypothesis that changing the pH of the algal samples would kill it was not supported because the control samples had decaying algae. The algae samples may have died due to the timing of nutrients or an unknown external factor capable of influencing pH.
When the pH of algae samples were adjusted once, it appeared that over time the algae was able to affect the pH level. Over the 3 weeks of measuring, the pH levels varied, but at the end of recording, only one sample, number 1, was out of a 7 to 8 range. Testing the pH in the future over a longer period of time, as well as using multiple different algal species and salinity values, is advisable. Increasing the density of algae to an area and testing the amount of time a manipulated pH takes to stabilize could provide significant findings.